Frequent lucid dreaming associated with increased functional connectivity between frontopolar cortex and temporoparietal association areas. What do we know about lucid dreaming? During lucid dreams, one becomes aware that one is dreaming while remaining physiologically asleep. It is verifiable through volitional eye movement signals. Variation in lucid dream frequency invites the question of whether the frequency of lucid dreams is related to individual differences in anatomical or functional properties of the brain. The prefrontal cortex, parietal cortex, and lateral middle temporal cortex show low regional cerebral blood flow throughout sleep. Hypoactivity of these regions has been postulated to underlie the diminished self awareness and volitional control during dream. Let us take a look at previous research done within this topic. An fMRI case study found increased Bol signal in many of these same regions mentioned above during lucid dreaming compared to non lucid REM sleep. EEG studies have also reported increased activity in the beta band over parietal regions. More research has found that APFC shows increased activation during self reflection on internal states. Patients with damage to this region frequently display metacognitive deficits such as inability to monitor disease symptoms. In support of a connection between metacognitive functions of APFC and lucid dreaming, a recent study found increased gray matter volume in two regions of the frontal pole in individuals who scored higher on a scale assessing the frequency of lucid dream. This research indicates that an analysis of brain structure and function in individuals who experience frequent lucid dreams while also controlling for dream recall frequency is needed. In the current research, we thus evaluated a sample individuals who reported lucid dreams spontaneously and a control group who reported lucid dreams once per year or less. The aim of the study was to test whether differences in brain structure or functional connectivity are associated with frequent lucid dreams while also controlling for dream recall frequency. A primary analysis investigated whether individuals who have frequent lucid dreams would show increased gray matter density or resting state functional connectivity of AFC. Here is a brief overview of procedures and setup with results, discussion, and critique. Demographic and behavioral results. The frequent lucid dream group reported significantly more lucid dreams compared to control group. No significant differences between groups were observed for working memory capacity or questionnaire assessments of mind wandering frequency, prospective or retrospective memory, or trait mindfulness. Voxel based morphometry. No suprathreshold clusters were observed for either the frequency lucid dream group contrasted with the control group or vice versa. No significant differences in gray matter density were observed for ROIs, and total hippocampal volume also showed no significant differences between groups. Seed based whole brain resting state functional connectivity. The frequent lucid dream group showed significantly increased functional connectivity between left AFC and the five clusters shown in the figure on the right. This group also displayed reduced functional connectivity between left AFC and the bilateral insula. There were no significant differences in functional connectivity between groups for right AFC. IPL-IPS subdivision analysis. We performed a follow-up analysis on the clusters in left and right inferior parietal lobe in order to characterize the overlap between these clusters and anatomical subdivisions of the angular gyrus. The cluster peak for both left and right parietal cortex was in the PGAs. From large-scale functional resting state network analysis. There were no differences in connectivity observed between groups within any LSN. The regions identified in our functional connectivity analysis overlapped with both DMN and FPCN, with the strongest overlap occurring within a subsystem of the FPCN. There were no differences in average network connectivity observed within this network between groups either. Results for whole brain graph theoretic analysis. No degree and strength showed significant differences between groups in left AFC with higher node degrees in the frequent lucid dream group compared to the control group, as shown in the figure on the left. No differences in between centrality or eigenvector centrality were observed between groups for any node. Here is a brief summarization of the main findings of the study. Our results converge with a recent fMRI case study of lucid dreaming, which found that a highly similar network of brain areas increased fMRI Bol signals during lucid dreaming compared to baseline REM sleep. Hypoactivity of these regions, coupled with preserved or increased activity in limbic or paralimbic structures and extrastriate cortices, has been postulated to facilitate a mode of brain function conducive to hallucinatory dream mentation. The current results suggest that increased functional integrity during wakefulness between APFC and temporoparietal association areas is associated with the tendency to have frequent lucid dreams. Dreaming and brain connectivity. It has been speculated that lucid dreaming is linked to neural systems FPCN. The FPCN is interconnected with both the DMN and the DAN. A recent study found that the FPCN could be fractionated using hierarchical clustering and machine learning classification into subsystems of FPCN-A and FPCN-B, which is connected to the DAN more strongly. Our results show that the frequent lucid dreams are associated with increased functional connectivity between APFC and a network of regions that showed substantial overlap with the FPCN subnetwork corresponding more closely to the FPCN-A. Further discussions on relevance of study. A meta-analysis of 120 neuroimaging studies of language and semantic processes found that the left AG had the densest concentration of activation foci across studies, with a significant clustering of activation foci also in MTG. The authors also noted that these regions are greatly expanded in humans compared to non-human primates, suggesting a role in the development of language. In our results, we speculatively propose that the APFC AGMTG network identified here may be part of the neural circuitry enabling the integration between heteromodal metacognitive and linguistic conceptual systems. Critique and further applications for our study. The measurement of individual differences in lucid dream frequency has been done in inconsistent ways and could be improved in future research. We used a questionnaire that does not measure variation in the degree of awareness of lucid dreams. Evaluating the duration of lucid dreams as well as degree of awareness would be valuable to relating brain structural and functional measures to lucid dream frequencies in future studies. Another limitation of our study is that our measurement of lucid dream frequency relies on questionnaire responses. This objective validation of individual lucid dreams in a sleep lab setting using volitional eye movement signals requires no protocols for physiologically validating the frequency of lucid dreams. This could be countered by attempting to physiologically validate at least one lucid dream in the sleep lab for each participant. We also did not observe any significant differences in brain structure between groups. This results contrasts with the study that found that two regions of APFC had increased gray matter density in high lucidity group compared to low lucidity groups. The authors of that study, however, noted that some of the results could have been partly influenced by differences in dreaming styles. This discrepancy in both the studies could have been an issue of statistical power, and it could be accounted for by conducting more research and addressing this question using large sample sizes to withdraw from conclusions. Future longitudinal training studies would be valuable in order to evaluate within subject changes in brain connectivity as a result of training to have lucid dreams, and to compare how such changes relate to functional network associated with frequent lucid dreaming identified. Another critique of our study is that there was no significant difference found between groups in working memory capacity or questionnaire assessment of prospective memory or trait mindfulness. This was inconsistent with the fact that sufficient level of working memory is required in order to become lucid during dreaming. We found that lucid dreams tend to occur spontaneously, thus it remains plausible that there could be a relationship between working memory and prospective memory and successful training in lucid dreaming. Another critique was that there was no significant difference in mindfulness in frequent lucid dreamers, and this is consistent with our research, which has found that outside of meditators, there does not appear to be an association between trait mindfulness and lucid dream frequency. Future work could also evaluate whether high frequency lucid dreamers show increased functional connectivity or bold signal during REM sleep. This would suggest that it may be possible to bias these networks towards increased metacognitive awareness of dreaming through techniques to increase activation of these regions. A recent study found that cholinergic enhancement with galantamine and ACHEI increased the frequency of lucid dreams. A follow-up to these findings would be to examine whether ACHEIs and galantamine may facilitate lucid dreaming through increased activation within the network of frontotemporal parietal areas observed here.